Yeah, is it working?
Yeah, that looks good to me. Do you want to try to advance your slide? I notice sometimes that can be glitchy too. That looks great.
Okay.
Okay, Robert, take it away.
Okay, thanks, Thad. Hi, everyone. Thanks for joining in this afternoon. It's my pleasure to introduce today's seminar speaker, Dr. Tadashi Fukami, Associate Professor of Biology at Stanford University. Dr. Fukami holds a bachelor's degree from Waseda University, master's degree from the University of Tokyo, and a PhD from the University of Tennessee, Knoxville. After earning his PhD, Dr. Fukami was a postdoctoral fellow at Landcare Research in New Zealand, then assistant professor at the University of Hawaii at Manoa. In 2008, he joined the faculty at Stanford, where he's been since. And his novel focus on historical contingency and community assembly has advanced the fields of community, ecosystem, and evolutionary ecology, with these accomplishments recently being recognized by the Ecological Society of America, of which Dr. Fukami has been named a fellow. His efforts also extend well beyond the field and lab and into the classroom, where his successful integration of research and teaching has helped promote inquiry-based education in ecology, garnering him the Science Prize in Inquiry-Based Instruction. Dr. Fukami, we're thrilled that you can join us today from the future. Dr. Fukami is joining us from Japan. We're about a day ahead, yeah? So yeah, we're great that you could join us, and I'm happy to, turn it over to you.
Okay, thank you very much, Robert. I hope everyone can hear me okay. Yeah, and so, yes, I really appreciate this invitation to come and talk to you and to give 2 talks. And yeah, I really wish I could come in person, but still looking forward to interacting with you, more of you later today and tomorrow. And because I was asked to give 2 talks, I thought about what to do. And then tomorrow, I'm going to talk about this the research that we've been doing over the past 10 years or so on the microbial communities that we see in flowers. And we have been using that as our experimental system to study what I'm going to call historical contingency in community assembly. And today, as a more of a general introduction to that field, a subfield in community ecology, how we think about historical contingency, I thought that I would give some of the historical background of this line of research by going back to some of the earlier papers, and then so it's not going to be so much about my original data, our group's original data presented to you. But today, I want to give some of the historical context. So that's why I call this today's talk, Origins and Frontiers, where we've been, we came from in terms of this research, and how that affects what we do today as at the frontier of this research is what I hope to do today. And I hope that it will be interesting to you. And so let's see if I can advance sometimes. Okay, and so to do that, I remember that I gave a talk, it's almost 2 years ago now, but there was a conference at the American Society of Naturalists where they talked about, celebrated 100 years of publication of the journal, The American Naturalist. Obviously that's one of the most, the main journals that ecology and evolutionary biology is founded on. And the idea of this symposium was for every speaker to pick just one paper that was published long ago in the journal, and then talk about how that influenced the research today and also personally. And so I was luckily part of this symposium, and I picked this paper called Multiple Stable Points in Natural Communities. And this is one of the foundational papers about historical contingency in community assembly. And so today's talk is based on what I gave 2 years ago, but I also updated some of the thoughts that I had since then. And so that's what I hope to do. And so I'm going to start with paper by John P. Sutherland. And he was a marine ecologist. He died young, unfortunately, but part of his one of the first experiments he did when he moved to Duke University, he did an experiment, a small experiment but that became important in thinking about influencing people thinking about ecological succession and community assembly. And so today, just to give you conclusions, what I wanted to do is to give these 3 points. So first, introducing this paper by Sutherland in 1974, I wanted to tell you a little bit about what this experiment was about and how that stimulated research in historical contingency. And then there was a follow-up paper almost 10 years later than that, but there was the Kono and Sousa 1983 was a very strong criticism of this paper by Sutherland. And I'm going to argue that this paper was also very critical that influences our thinking even today because it was because of the heavy criticism of this paper by Sutherland that caused the research to remain a little bit dormant in terms of thinking about historical aspect of community assembly, but also at the same time, kind of paradoxically, this paper also, in my view, stimulated and broadened the research that we do today in historical contingency. And then based on that, I am going to say that there is much more to be gained by studying more about what makes communities historically sensitive. And so that's the point of my talk today and the points of my talk today. So getting back to Sutherland experiment. So he got a faculty position at Duke University Marine Lab. And then when he went there, he did an experiment, a simple experiment where he had a small tile submerged in just outside this marine lab that the Duke University has, and then just waited to see what kind of fouling communities develop. So these are bryozoan tunicates and those ones that we often see in the sea. And he submerged these experimental small tiles at different times of the year and just observed what happened and expecting that there's a nice orderly succession happens. But instead, what he found is that depending on the season in which he started this small marine community, things were very different. So in some cases, there are 2 species of these tunicates that became very dominant, but in other cases, when it was the tiles were submerged at the different times, it was a completely different species that dominated the community. And I think he was a little bit puzzled by that because still at that time, the dominant view in community ecology is that there's a expected succession that happens. And so I think he was having a hard time trying to summarize the results for publication. And then he has this kind of recollection of what happened. And so I wanted to, it's interesting. So I wanted to just give you this actual sentences that he had. And so he said that he was visiting Steve Hubble at the University of Michigan. This is the neutral theory community ecologist that's now very well known, as you know, and still active. And Steve, and he was, John Sutherland was saying that Steve was office mate in grad school at Berkeley and had invited him up to give a seminar. And while discussing my results that he had from Duke, he said that you are not looking at classical succession. You've got evidence there for instead for alternative steady states. That's going to be one of the keywords here in relation to historical effects. But anyway, here read this paper by Lewontin and see if you don't agree. Indeed, Lewontin paper did outline the appropriate theoretical construct within which to view not only my results, but the results of many others. And I returned to Beaufort and over the next several months wrote the paper that I sent to American naturalists. Why not go for the gold? And so this is the paper, resulting paper, once again. And you can see that the very first word that he used was Lewontin, citing Lewontin. And so this paper is this publication that he cited by Richard Lewontin. It's a little strange because he's more of a population geneticist, but this small paper that he had in this obscure symposium proceedings became one of the theoretical foundations for thinking about historical continuance. Even today, this symposia proceeding gets cited. But what he was saying in here, this is a completely theoretical paper, saying that often when we think about stability in ecological communities, we think about just stability and that's it. But there's actually important distinction between what would be considered local stability versus global stability. The communities might be in a stable state, but that could be in a theoretical sense a local state, because there could be an alternative local state that's also stable. And that's different from thinking about global stability, meaning that if you have this set of species that you construct your communities, and if you have some certain conditions, then there's only one stable condition that all communities get to, regardless of the history. It can be different because there are multiple domains of attraction. It's all very abstract and theoretical, but there could be multiple domains of attraction depending on where the community is started from in terms of the initial species composition and abundance. The communities can go in different trajectory to end up in different alternative stable states. And so so John Sutherland used that as a theoretical basis to present his experimental results from the marine communities that he studied and explain that these strange things that he's observed in terms of communities doing very different things depending on the initial condition can be considered in relation to what Lewontin was saying theoretically. And so in my view, I think that the Sutherland experiment is like Gauss's experiment for competitive exclusion principle, meaning that as most of you probably know, Losca Volterra, those are the people early days who try to establish mathematical models to explain what we expect in terms of resource competition when there are different species competing for for same resources, and there was this competitive exclusion principle. But that was, even though the model was based on some specific problems of a fishery, the model itself was abstract. And as an ecologist, at least me, I'm much more of an empirical person. So it's not very intuitive when you're presented with mathematical models. But then the Gauss in Russia did this experiment with paramecium, showing that with a concrete species, that you can do experiments to say that what happens in mathematical world can be demonstrated empirically with these experiments that is much more intuitive because it involves real organisms. In the same way, this historical contingency, the fact that the communities can develop in different ways depending on initial condition. The Lewontin's paper is really abstract, and if you read it, it's at least I have a hard time understanding fully. But then this Sutherland experiment, when he published this paper with real organisms, I think that made it much more accessible for many people to think that maybe this idea about historical contingency is an interesting thing that we can actually study with real organisms. And so I think that's the role that the John Sutherland's experiment played and popularized the idea. So now I'm using this review article that I was also involved in. You can kind of graphically summarize what I was trying to say. And so in a classical succession idea, it depends on the habitat. And so if you have a different physical and chemical conditions in one local place as opposed to other, then you can use that information about habitat to predict what's going to happen in terms of species composition that you expect after some community assembly happens after some disturbance. But then there's this alternative scenario where even if you have the same habitat condition and same species pool, same species in your species pool, then even just by having some variation in which species came first and which species come later, often by chance, this immigration history can have an overwhelming effect on what you see in species composition, and it's also called priority effects. So it's an interesting thing because often when we see variation that we cannot explain in terms of community structure, like species composition and diversity, when we compare different communities, we, as community ecologists, we often have to give up. This is something that's not distinguishable from random expectation. So it must be stochastic, and that's it. That's the end of the story. We have some stochastic variation, and then we need to focus on other things that seem deterministic, like physical condition of the habitat, to explain community assembly. But that's not always the case, because as soon as you look into historical background of the different communities, there may be some hidden mechanisms. So citing this my former my PhD advisor's paper, this remains to be one of my the favorite papers. There is a quote here that the community can appear idiosyncratic when they're actually not, because when you look into history, there could be some mechanistic explanation you can give. And so now I think one of the active overarching question that I think is productive to address is summarized in the title of this paper by John Chase that's published in 2003. When should history matter? It's often difficult, if not impossible, to go back in time. We don't have time machine. So if we don't have data in terms of these detailed historical data, in terms of which species came first and which species came later, if we don't have that information, we kind of are at a loss, because if that's important, that those kind of historical contingencies are important, and if we don't have historical data, we don't have a way of explaining it. But instead of trying to predict each specific community, another question you can ask is this one: when should history matter? Are there conditions that we can use to predict how sensitive communities might be? And is there other conditions, physical and chemical conditions, or maybe something about the traits of species that you have in the species pool? Can you use those data that are that you can collect to have some systematic expectation of when should history matter? Or in other words, when should we expect multiple stable points, the kind of phenomena that John Sutherland saw? And so in this paper by John Chase, which is also still a great paper, very relevant today, what he did was to look at different ponds. And so there are 10 different watersheds, and each watershed had multiple ponds, and he just looked at the diversity and composition of aquatic plants and insects. And then so that as you increase, as you go to different places with different productivity level, some are nutrient rich and some are nutrient poor, there was this increase in dissimilarity. And he thought that this increase in dissimilarity with primary productivity could be because each community are more likely to go into different trajectories if the productivity is high, because of stronger priority effect there compared to low productivity ponds. And because of that, there is an increase in dissimilarity, which would lead to different patterns in species richness. If you look at the local community, as you increase productivity, old ideas say that that would make the competitive interaction, other species interaction strong, so that eventually there's a decline in local species richness. But then if you look at regional species richness, the pattern can be completely different because of this increase in dissimilarity with a stronger priority effect. As you increase productivity, the regional species richness can continue to increase. So this difference between regional and local species richness that we see in panel B is because of the stronger role of historical contingency. And so what John Chase was saying in this paper is that by asking this question, when history should matter, there may be things that we can do to explain patterns in communities like species richness that we could not otherwise do. And so I think answering this question is good because it contributes to understanding community predictability. So when should we expect communities to be more predictable because they are not sensitive to history, and when they are more sensitive to history and predictability being more difficult, that itself is a good kind of goal, scientific goal that we can think about. And then as the second thing is that is beta diversity. When we think about biodiversity, it's not so much just about local diversity, alpha diversity that we are interested in. It's also the overall regional diversity, larger scale diversity. We are also interested in understanding. And if the priority effects and the historical contingency can be a source of beta diversity, like in the John Chase's pond experiment, this historical contingency and when that matters can be something that we haven't looked into so much that can be helpful in understanding what controls beta diversity. And then third thing is that answering this question, when should we expect multiple stable points, can be also useful from more applied perspective, because for example, when you're trying to restore some community, for example, you could choose to reintroduce different species at once, or maybe you can also do that sequentially. And if the priority effects are important, so that the reintroduction order matters, then you want to be so careful about that. On the other hand, there may be cases where the introduction order doesn't matter so much. In that case, you can expect restoration to be more successful. And also in the same way, it can be applied to agriculture when you trying to have biological control, or it could be in more recently, there's a lot of interest in human-associated microbial communities. And then, for example, gut microbial communities, as you know, are complex, but there are also strong species interaction going on, and still speculative at the moment because experiments are hard to do with humans, of course, but these priority effects could be also important in thinking about how to make the gut communities, microbial communities, at a healthy state. And should we think about a historical contingency and when? So there are some applied implications in trying to address this question. And despite that, when you look at the literature, I would argue that this historical contingency remains to be a really minor component of community ecology research. And so if you look at this is a review article that I had now 5 years ago, but I think the general trend remains the same. What I did was to look at papers that were published on community assembly, and there's a lot of increase in the number of articles like you see in panel A. But when you look at what these papers were about, and I used different keywords. And so, for example, functional traits or phylogenetic patterns of community assembly, or trait-based ecology and priority effects and so on. Compared to those other common key terms, the papers that were about priority effects, I also used alternative stable states, multiple stable states, and regime shift and hysteresis and those other related words. But despite that, the proportion of papers that mention priority effects remain really low, like less than about 10%, indicating that the historical contingency is something that I think is thought of as inconvenient thing, because if we don't have historical information, the research cannot be productive. I think that's still the kind of common sentiment that the community ecologists have. And I would argue that this question that were put out by John Chase, when should history matter? It's not just him, but some other people have argued for that. This question, when should we expect alternative stable states or multiple stable states? I think there's a lot to do with that. And and then finally, I get to this paper that I mentioned by Connell and Sousa. As I mentioned, this is a paper that criticized heavily the Sutherland's paper that was published in 1974, saying that actually this experiment didn't provide enough evidence, strong enough evidence to say that he saw multiple stable points in his communities because of these three reasons. One, you would have to look at observe communities over multiple generations. Otherwise, it's trivial because you're just looking at individual response to other individuals, and it's not really community assemblies. It's more of a behavioral or some physiological thing that you're looking at. And so because Sutherland's experiment was too short to meet this criterion for judging stability or showing stability, that experiment wasn't really good, is what Kono and Sousa said. And then second, you have to establish that the abundance of different species are relatively constant over time, because that's about stability, and it's related to the first criteria. Because the experiment was too short to think about multiple generation dynamics, once again, Sutherland experiment wasn't really providing evidence for multiple stable states. And then thirdly, they also said that you have to look at communities that are under same physical environment. I think in Sutherland the experiment, this was okay. This might be okay, but in some cases, when we think we found evidence for multiple stable points, it might be just because these communities that we are comparing are experienced some temperature differences or something that's outside of what happens in communities. It's more of external environmental conditions that led to different communities. In that case, it's a little bit obvious because in a different environment, you expect different communities. It's not so much about sensitivity to initial condition. And so this was a really strong argument that was made in this paper. And I think that made the Sutherland paper not as appreciated otherwise. As you can see, we can only speculate what happens here just by looking at number of citations. But what I see here is that the Sutherland paper gets cited, used to get cited a lot, but when the Kono and Sousa paper came, that was cited a lot and talked about. And then it seems like there's a decrease in the number of citations by Sutherland. And I think because of this heavy criticisms by Kono and Sousa, I think this research on multiple stable points, I think it's possible that it's considered not so productive. And then I think that might have led to the pattern we see today, that there aren't too many papers on historical contingencies even today. And so what I wanted to do for the rest of the time I have is to argue that after this Kono and Sousa, there was a slow but steady increase in the reappreciation of multiple stable points as an interesting phenomena to study. And also at the same time, by different people trying to respond to Kono and Sousa's criticism, it resulted in expansion of the question about historical contingency. And so getting back to this question, when should we expect multiple stable points in natural communities? What are the conditions that we can think about to expect a stronger or weaker historical contingency? What I'm going to do is to say that there are four things as an extension of this question when multiple stable states happen. And so one is it's not just about ecological phenomena, but also evolutionary phenomena that where historical contingency can be relevant. And it's also about not just the stable points, but also the transient states, transient points, or I guess transient dynamics that can be also considered just as equally or even more relevant in thinking about historical contingency. And then thirdly, in addition to stable points, we can also consider what you might call assembly cycle as also something relevant to historical contingency. And then the last one is instead of just thinking about community structure like species composition, and species diversity. It's also interesting to think about the functional aspects of communities, the ecosystem level patterns in communities and in relation to historical contingency. And so what I wanted to do is to go over these 4 points quickly to see how we ended up expanding this question by thinking about these additional things. And so getting back to this criticism, they were saying that that you have to look at many generations until you can be sure about stability of communities. And that's summarized in this quote that I have here by Robert May. There was a paper in Nature in 1977, and he said that if there's a unique stable state, historical accidents are unimportant. If there are many alternative local stable states, historical accidents can be of overriding significance. And so he was also interested in historical effects and alternative stable states. But it's clear that when you look at the community assembly long enough, and if they get to the stable states, and if there is no effect of history, then history is not important. And so that's about that's a heavy emphasis on endpoint, the stable point. But what I would argue is that that's a little bit too narrow in terms of considering historical contingency. And to make that point, I'll just use one of my really old experimental results that I have. As a PhD student at the University of Tennessee, I used these protists like ciliates and rotifers to do experiments. And I was manipulating assembly sequence. And so I introduced about 16 species of these microorganisms in different order. And I was manipulating ecosystem size, just these different size of different jars of different size. And what I found in terms of species diversity in these microbial communities is that if you have a small jar or small vial, then in terms of species diversity as an aspect of community structure, the assembly sequence seems to matter, because in some cases, even though other conditions were all identical, in assembly sequence, let's see, C and D, if you look at the ecosystem size 5 milliliter, the diversity is lower compared to when I assemble the same communities just in different order, sequence A and B, and high diversity, low diversity. But that didn't seem to happen if you have a large container. And I have reasons for why that might be. But the point here is that if you run this experiment, and by the way, these dashed lines are the timing of the introduction. So there are 3 different introductions that I used, but in different order. And so, but the point is that here we see that it seems like the alternative stables happen if under small ecosystem size, but not in a large ecosystem size. And I was happy because I was interested in these alternative stable state. But then I thought that I should run this experiment longer, and found that the kind of historical effect disappeared. And so according to May and other people at the time, my conclusion here would be that the history is unimportant. It doesn't matter what history you have, because they all get to, at least in terms of species diversity, they all get to the same level. And so this is a very historically insensitive community that we see here. But I thought that was a little strange, because in this case, the species that I used, these microorganisms, had a generation time of 1 or 2 days, or could be a little longer, but could be shorter. But when you look at the time scale, the 40 days or so, a month or so, corresponds to many generations. And so even though when you look at the stable point, endpoint, the history doesn't matter, when you look at the transient state, transient dynamics, the history mattered for a long time. And when you look at, this is just a small microcosm experiment, but when you think about tree communities or any communities in the field, often what happens, as you know, is that the disturbance happened frequently enough that most communities, I would argue, don't get to stable points. And so thinking about this long-term transient states can be actually more relevant in understanding ecological communities than focusing on stable points. And so, this transient state is something that I think we need to think more about. And then here I did, this is just a computer simulation paper, and I was showing that when we look at plant communities with that's affected by plant soil feedback, you can also see that depending on what scenarios you have, I don't go over details, but this is an example where when you look at beta diversity, that's resulted from historical contingency like priority effects. If you run this computer simulation of plant succession long enough, there's no difference between these two scenarios. But before getting to that stable state, there is a difference between these two scenarios in terms of beta diversity, showing that at the transient state, long-term transient state, there could be historical effects, even though you don't see that eventually. And this is a more problematic case where if you compare different scenarios, one scenario might have a higher beta diversity at the end when you look at, when you focus on stable points. But along the way, the other scenario might show higher beta diversity as a result of stronger priority effects. So that your conclusion about when to expect historical contingency can be opposite depending on whether you look at the stable points or transient states. And so I think that there's much more to be done in terms of transient states here. And then I have 3 more things, but I think I should go over these quickly in terms of time. But another thing I wanted to say is that there's also this issue of assembly cycles, meaning getting back to this criticism, Colonel Sousa was saying that you have to make sure that the abundance stays constant over time to say that the community is stable, and that makes sense. That's intuitive. But when you think about historical contingency, it's not just about stable points that can be interesting. It can also be some fluctuation that can be sustained over time that can be interesting in relation to historical effects. So for example, here we see that in some of Sutherland experimental communities, it seems to be there's a coming and going of different species. And theoretically, what you can do is to do a local kind of a common simple local Volterra communities of trophic interaction, predator prey, and different multiple prey species and predator species, and show that there can be a permanent, what's called permanent end cycle, where you can have a specific community species composition at a time as a result of this sequential introduction of species, at least in the model. But then to get to a different state, there is only certain species, for example, species 1 here, if you look at the this arrow with 1 going to it, and then only one of the species in the species pool can move the community to another state, and there's another, and then there was a species 22 that's the only species that can invade that specific community, and then this cycle can go on forever, at least theoretically. And so when you think about effect of immigration history, this is relevant because depending on what immigration order you have, at a given point in time, you can have variation among communities. If you have this specific history, it could be one of those 4 states that the communities stayed even after some long time. And then if you look at another community, if they have different history of species immigration, they can be in a different state, in a different state within this what's called permanent end cycle or assembly cycle. And so if you're here, you cannot get to these points unless you move from through this trajectory. And this is all abstract and theoretical, but there was Chris Steiner did an experiment with a zooplankton in a microcosm once again, because the microcosm is always the first step to test some theoretical ideas, and you can actually manipulate the species immigration order and see what happens over multiple generations. So in this time in zooplankton, what he did here was to introduce these different species of like Daphnia and other zooplankton at once, simultaneously. He was introducing different species, and he used the same set of species, but introduced these species sequentially. And what we found is that even when the sequential dispersal was used, then there was a constant turnover of species. And depending on which order of sequential immigration he used, communities were going through different state. And so when you look at beta diversity here, if you have a sequential dispersal, then and if you manipulate at the same time nutrient concentration, productivity of these experimental communities, the beta diversity, the high beta diversity as a result of this assembly cycle was higher under higher nutrient concentration, showing that even though we don't have multiple stable points here, because of this historical effects that happen as a result of assembly cycle, we can get some ideas about when history matters. When in this case, higher productivity or higher nutrient concentration is the condition where you expect greater historical contingency. Okay, so that's about assembly cycles. And then the last part, the third part is I also wanted to mention function of communities. And so this is related to the third criteria that Kono and Sousa had. You have to look at compare communities under the same physical environment to test for the existence of multiple stable points. And this criticism itself was criticized by some others. And so particularly in this paper in 1984 by Charles Peterson, who is another marine ecologist, he questioned this criteria as something that's useful. And his point was made by this specific example of his study system that he had. And here we have two species of bivalves. They look the same to me, but they're different species here. And so in the marine sediment, often you see that in one local area, there is this one of the two bivalve species that are dominant. In other area, there is the other species that are dominant. And you can see in this plot that it's either of the two. There's no co-dominance here, and the explanation here was that one of the two species of bivalve is associated with a ghost shrimp that bore into this sediment, and what shrimp does is to change the physical environment so that it's more favorable to this the bivalve species that are commensal with the ghost shrimp, because they live with this shrimp in the burrows that make, and that kind of physical environment that the shrimp makes, it's like an ecosystem engineering, is not conducive to the other bivalve, and because of that we have this alternative stable states. And if we think of this as a case of alternative stable states, you cannot explain this historical effects. So in here, whether you happen to have shrimp initially abundant determines which species of the bivalve will become abundant. And so this phenomena you cannot explain without thinking about the changes that are biologically induced changes made by ghost shrimp in terms of this sedimental structure. And so if you exclude physical environment, different physical environment when we think about alternative stable state, that's too restrictive, is what Peterson said. And since then, there's a lot of examples. And so my favorite example comes from James Estes' work on with kelp and urchin dominated system. If you go to these different marine communities, what you see is that there are places where sea urchin is very abundant, and there are other places where kelp is very abundant, but you cannot have both at the same time. There's no codominance here. And with experiments that they have conducted over years, they found that there is a positive feedback, so that once urchin becomes abundant, because historically they were a little bit more abundant than kelp, there's a positive feedback so that the urchin becomes more abundant, and that makes the environment more difficult for kelp to come back and become abundant. It's the same way when you have kelp, because of the wave action and everything that kelp does, that makes the habitat very challenging for urchin to come back and become abundant. And so this seems like another case of alternative stable states. But the point in relation to this physical environment is that this two alternative stable states is associated with a host of different physical differences that result from the interaction between urchin and kelp. For example, they studied the carbon dynamics and showed that depending on which of kelp or urchin, which one is more dominant, the carbon sequestration can be completely different. And so it's not so much just about community structure, but also the functional aspects of communities like carbon dynamics and in the case of shrimp, the sedimental physical structure that can also be different as a result of historical differences in initial community composition. And to introduce you to just another example, this is the example study system that I'm going to focus on tomorrow. So this is just a kind of a preview of what I'm going to talk more about tomorrow. But what we found in these microbial communities that develop in floral nectar in different flowers, what we found is that these microbial communities in nectar can be dominated by either fungi, the yeast, or the bacteria. And so once again, we have a L-shaped pattern here that is either, so these different points represent individual flowers. And so if you look at one single flower, you can have either yeast dominating or bacteria dominating, but not the other. And I am going to talk about different data indicating that this also represents alternative stable states. But in this case, in terms of function, we also found that the reproductive success of flowers can be different depending on which is dominant, yeast or bacteria. And so it's not so much just about microbial community composition, but also the function of the habitat, in this case, the flower, the seed set and everything that's critical for plant population, that can also be affected because there is a physical changes to the nectar chemistry, chemical changes, and also sometimes physical changes like a temperature can also be affected as a result of yeast population dynamics. And so there's a lot of functional differences that we can expect. And so this is another expansion of this research on historical contingency. And lastly, I wanted to end by thinking about evolution. And so there was this paper that was published in 2002 by Demister and others, showing that this is just a single species dynamics, population dynamics, by looking at particularly the Daphnia population that they've been studying. They argued that there can be a rapid evolution that can happen in different ponds where Daphnia populations are found, so that the Daphnia can rapidly adapt to the local conditions. And once that happens, the other genotypes, the genetically different individual that might come to that pond later, have a harder time to join that population, because now the population is more adaptive than the newcomers. And so that within species, this is just within species, but there can be priority effects that happens as a result of this rapid adaptation, local adaptation. And so previously, as a community ecologist, we assume that evolution is something that we can ignore because it takes long time for evolution to happen, adaptation to happen. But it seems like the evolution we should also think about when we think about priority effects and colonial invasion resistance. And so later, they extended this idea at the community level to say that even when you look at different species interacting, this local adaptation idea where the early arriving species can go through rapid local adaptation that can enhance this preemptive priority effects. And here I'm just showing one of the experiments that I did some long time ago now. But here we have.
You're welcome. Yeah. No. Sorry, I was in.
Okay.
Can people make sure to turn their mics off? Thank you.
Yeah, thanks. Yeah, so I should wrap up soon, but do I have a few more minutes, Robert?
Yes, yeah. I think, we can go past 5 a little bit if you're okay with that.
Okay, yeah, I try to finish by 5 for sure. Yes, okay. Thanks. Yeah, and so in relation to evolutionary questions, I wanted to just share this result of experiment. It's not so recent, as you can see, 2007. But what I did here is I used the bacterial population of the Pseudomonas fluorescens, which was known to undergo very rapid diversification, as you can see from panel A, B to F here, that if you look at number of genotypes that you see after you introduce ancestral isolate to experimental vial with a rich nutrient media, it's previously known that they go through rapid diversification. But what I found in this experiment is that if you use 2 different genotypes, the names here don't matter so much. You don't need to understand what these different names really mean. But the point here is that I used 2 different genotypes that are ecologically different in terms of the habitat use in this vial. And so what's called small SM and small WS. And I manipulated the relative timing of the introduction of these 2 genotypes. And what I found is that in terms of the extent of diversification in this bacterial microcosm, it was completely different depending on what order of introduction that I used. And so if this purple one came earlier, then they can undergo extensive diversification. But if that genotype was the late comer, then because the other one doesn't go through diversification, but also very competitive against the other, that the suppression of the other population that would otherwise undergo diversification is not so successful. And so that results in absence of diversification. So in terms of evolutionary diversification, the historical effect we can see, this would be hard to observe, of course, in plants or animals because it just takes too much time to observe diversification. But you can also do some analysis showing that if you look at the phylogenetic patterns of different clades of plants, and so here what I'm showing is New Zealand Alpine plants, and in New Zealand it's still challenging, but you can have some idea of when different clades, different genera came from outside New Zealand to colonize New Zealand. And you can look at the estimated arrival time of different clades and use that to see whether extent of diversification. So you can look at the number of species in each genus to see whether there is a relationship between the time of their arrival in terms of evolutionary time, phylogenetic time, and the extent of and the number of species within each genus, and there is a positive relationship, so that the older ones are more likely to be more species rich. This is just very indirect evidence, but showing that maybe something like what I found with this bacteria could potentially happen with plants, and there is also indirect evidence from, for example, lizards in Caribbean, anolis lizards from Caribbean islands. And so the idea seems potentially applicable to other organisms, not just bacteria. And I'm also going to, I hope to talk tomorrow about the latest, this is unpublished results, but my graduate student, Kelly Chappell, she did an experiment that's also related to this idea of evolutionary priority effect. So I hope to talk about that tomorrow. But then I quickly go over these 4 points. But what I wanted to say is that despite this trend in the literature, where the priority effects and other related topics that are related to historical contingency in community assembly, despite those papers remain to be a minor component of the community assembly literature, there is steady increase in this realization that the history is something that we can actually embrace or study, and that can lead to a deeper understanding of the community patterns like diverse species diversity, composition, and even functioning of the communities. And so that's what I wanted to say here. So this is the points of my talk today, and hopefully it was coherent, and I hope that I can continue this topic with a little bit more specifically using the nectar microbes as example of what you can do to study historical contingency empirically. Okay, thank you very much. I'm not sure if there's time for questions, but I would appreciate any question you might have.
Great, thanks, Tad. Let's give Tad a round of applause. And I think, yeah, given maybe the number of participants here, I think if people, if a few individuals want to turn on their mic in their video and ask a question directly to Tad, feel free. I can also monitor the chat and relay